Hello and welcome guys to the another episode of our discussion series. Today I'm going to have a great conversation with one of my dear friend Kanchan on the topic of vaccine and drug development. And this is really a good time because nowadays many people are confused about COVID-19 vaccine and also its side effect. So I thought this would be a great content for my YouTube channel. And also I'd like to add that Kanchan is one of the significant contributor to my YouTube channel. He helps me develop a script, he helps me edit video, and also helps me upload content to my YouTube channel. It is because of Kanchan I can upload video in my YouTube channel every week. So thanks a lot, Kanchan, for that as well. So let's welcome Kanchan and let's learn about vaccine from his expertise and his experiences. So Kanchan, thanks a lot for being here with us today. Before moving directly into the topic of vaccine and drug development, would you like to introduce yourself to our viewers?
Thank you very much, Biso. Hello, guys. I'm very glad to be part of Inspire to help Biso develop contents. And also, I'm very glad that you gave me an opportunity today to uh, give my few thoughts about drug development and also a little bit about vaccines. To introduce, I am a research scientist currently working at Structural Genomics Consortium, Department of Pharmacology and Toxicology at University of Toronto, Canada. For the past 7 years, I have been working mainly with developing and testing drug candidates in vitro and in vivo for various targets, such as for diseases such as cancers, neurodegenerative disorders, and also recently I have been involved with developing antiviral drugs for COVID in proteins as well. Uh, so I normally develop assays to look for molecules that inhibit or either stop the function of these enzymes.
Thanks a lot, Kanchan. That's great. So how long you have been involved in working as a researcher for drug development and vaccine development?
My primary research has already, I did my PhD in biomedicine from Department of Drug Design and Pharmacology at University of Copenhagen. And since then I have been heavily involved with designing drug-like molecules,
Yeah.
not exactly drug, but I'm more involved in preclinical studies for these molecules. So what we do is like we try to design certain type of molecules or certain drug-like candidates that can eventually pass on for clinical trials and eventually be done as drugs. I haven't really worked with vaccine development, but I have been following it lately a lot because also we are working with developing antiviral drugs for COVID targets these days. So
Yeah, that's good. So let's talk more about this vaccine and drugs. So can you give us overview about vaccine and drugs in a simple term so that everybody can understand?
drugs are, to put it simply, so drugs are generally administered to treat when you have already have a problem. So if you have a fever or like if you have a stomach ache or headache or something, or if you have any other disease, you take a drug or like basically
Yeah.
medicine for it. Whereas vaccine is something that you normally administer to prevent the appearance of this problem.
Yeah.
So if you have a vaccine for COVID, SARS-CoV-2 or COVID, what it
Yeah.
means is like you want to protect yourself from not getting it in the future.
Yeah.
Whereas if you have a drug for COVID, it means that once you get the disease,
So basically, vaccines are given before the disease happens,
yes.
and drugs are given after the disease. Yeah. So let's talk more about these vaccines development procedures. So I think our viewers would be interested to know how these vaccines are developed, how scientists develop vaccine from scratch. So can you please give us a little bit overview on how we develop vaccines?
As I said, I am mostly working with drug development, but vaccine development is also like the same approach and like the similar time frame.
Yeah.
Normally, in normal sense, what happens is like we do a lot of preclinical trials, like a lot of research happens in
Yeah.
different research labs of universities, at pharmaceutical companies, and different research institutions. So this process has been going on, like this goes on even when you know there's a problem, people start working on it, and then there are certain candidates that
Yeah.
you think like, okay, yeah, this could work. So then you do a testing on in vitro testing. What this is what we do it on the bench scale. We try to mimic the human body or like system, and then we try to do the experiments in the lab bench, and
Yeah.
then it moves to the clinical to the cell phase, in vivo phase, where you do it on the cells.
Yeah.
Like you take the cancer cells or some other disease cells and
Yeah.
you try to see if the molecule works. Then once this passes this preclinical trials, or I would say once it passes this phase,
Yeah.
it goes to clinical phase or clinical trials, and
Yeah.
it usually involves 3 or 4 different kind of steps.
Yeah.
So the phase for the first phase is usually to determine if it's safe to give the vaccine or drug to the
Yeah.
person, like whatever molecule or whatever thing anybody identified,
Yeah.
and how much amount to give it. So do you need 1 milligram of it or like half a milligram of it? So basically, the first phase involves trials to determine is it safe and what is the
Yeah.
dose required.
Yeah.
In the second phase, what you do is you try to get an efficacy rating. So there's a
Yeah.
difference between efficacy and efficiency. Efficacy is like under optimal conditions. So like in a regular condition, what would be the effect of this drug or
Yeah.
vaccine? So like
Yeah.
how good it is. Then
Yeah.
you do it in a small number of patients, like having the disease, and then you explore the safety of it in those small trials.
Yeah.
Then you move on to phase three trials, which are
Yeah.
large, like large trials,
Yeah.
where you again determine the safety and efficacy in very large various number of patients, like with
Yeah.
different like diversity as well. And then once it goes there, you advance to the new drug application stage, and then once it passes the phase three, it goes to phase four, which
Yeah.
is when it is approved by regulatory agents like the FDA, and then
Yeah.
you do post-market surveillance studies for it, like how effective it is, what are the side effects even after 5 years, 10 years, this thing goes on. So normally a process of drug design or vaccine design usually takes from 10 to 20 years
Yeah.
in a normal speed.
So you just said normally it takes 10 to 20 years to develop a vaccine. But this COVID-19 vaccine, it has already been in the market in less than a year. So what is the change in that procedure? This vaccine has been developed at such a fast rate.
So that's a very nice question. It's very interesting as well. Normally it takes 10 to 20 years, but what happened here with COVID is it was a combination of different kind of factors, I would
Yeah.
say. First of all, it's a very good example of what science can do very quickly if it's allowed to, like if we are allowed
Yeah.
to, what we can do, right? But it
Yeah.
did not happen overnight. There were research going on for different kind of coronaviruses that we already saw before, SARS and MERS as well, Middle Eastern Respiratory Syndrome, and
Yeah.
then the SARS-CoV-1 that appeared in around, I think, 2002 or 3. So
Yeah.
people were already working on a concept to develop vaccine for these kind of coronaviruses. And also, what happened was like there was this Ebola and Zika virus breakdown as well. So people researchers and even the regulatory bodies all over the world, they united together. They said like, well, we need to form like a national or global infrastructure
Yeah.
that can promote faster vaccine development. And also
Yeah.
these regulatory bodies, what they did was like they shortened the time frame that is usually required. So for
Yeah.
example, if it takes 6 to 7 years to finish all the phase one, two, three
Yeah.
of this design or vaccine development for the COVID case, everybody united and then they did it together and they reduced the time needed as well.
Yeah.
So also what helped here, what I think is when you have like a virus outbreak or some other outbreak in very localized area, you don't
Yeah.
get enough participants to try the
Yeah.
vaccine. But SARS-CoV-2 spread it globally so fast
Mm.
that it allowed researchers actually to have trials for vaccines in various demographic settings and various kind of people, different
Yeah.
age groups, different symptom-wise people are in like more temperature-wise and
Yeah.
also various demographic settings. So what it allowed was you could do the phase three trials and everything very fast as well. And
Yeah.
yeah, uh, this was very well done and is the one that developed Pfizer vaccine. For example,
Yeah.
Genentech was already working, BioNTech was already
Yeah.
working on this concept for more than 3 4 years. They were
Yeah.
working on messenger RNA vaccine, which is
Yeah.
the first thing actually, which is very good for science.
Yeah.
Because before this, there was no DNA or RNA vaccine,
Yeah.
and they were already working on this technique.
Yeah.
And once they said that okay, yeah, they basically within a year, I would say that it was approved. So this is a
Yeah.
big record as well. it has actually established that now messenger RNA or DNA can also be used or granted for use in people for curing disease or for curing vaccines.
Yeah,
And then also it's very easy to make compared to other different types of vaccines we have. And
yeah,
then it's also less complicated to make, like, or to
yeah,
administer compared to making proteins in cells or making live virus. So it simplifies the manufacturing,
yeah,
and you can use the same facility or same thing for other viruses as well that might appear in the future. So this
yeah.
approach is very nice. I think the scientific community did a very good job.
Yeah.
With
So you just mentioned messenger RNA vaccine and DNA vaccine. So what are the other types of vaccine that are available and that were used previously in the world, and now you are talking about mRNA? So can you please give us overview about different types of vaccines out there?
so normally what the vaccines we receive for like virus or bacteria for the infections is you can have a live attenuated version of it. So this is attenuated means the actual virus, but it's a
Yeah.
weakened version of it. You
Yeah.
could so it could
It's
stimulate your
whole virus, whole virus.
yes whole
Yeah.
virus, but a very weakened version of it,
Yeah.
so that it will stimulate a immune response, but it
Yeah.
will not cause you serious disease, right?
Yeah.
And then, but then this also another approach could be like you could use an inactivated vaccine, so
Yeah.
where you could like so the vaccine, so the virus, for example, or the organism is already killed by chemicals.
Yeah.
So it's safe to use because organism is already dead. But then again, it may not be as effective as the live virus. Another approach was also you could use subunits of a virus
Yeah.
particle, for
Yeah.
example. So you use a piece of the virus, for example, how it attacks or a part of it, part of its body.
Yeah.
So you
Yeah.
could focus, you could teach your immune system how to react to it if it
Yeah.
comes to you, right? So
Yeah.
and another one was also you could use a harmless virus, like a
Yeah.
virus that is not harmful for you, and then you
Yeah.
could
Yeah.
deliver the genes from this harmful virus to the
Yeah.
harmless one and then
Yeah.
take the virus
Yeah.
yourself. But these approaches have been there already. For example,
Yeah.
the attenuated version has been there for chickenpox or
Yeah.
measles, and
Yeah.
the inactivated form has been used for, I guess, polio. And recently, before SARS-CoV-2, the vaccine that was developed was for Ebola, was using the viral vector. So now what they did was they used the DNA or RNA molecules. So it's
Yeah.
very easy and also quick to design, but
Yeah.
like it has never been, it was never done before
Yeah.
because, and also there were no licensed DNA or RNA vaccines before. They were
Yeah.
thinking it is too harmful to do it or all these other hurdles, but
Yeah.
luckily, we have here Moderna and also Pfizer vaccines that using the messenger RNA of SARS-CoV-2.
So, Kanchan, this COVID-19 vaccine, let's talk about this COVID-19 vaccine. So what are the different type of COVID-19 vaccine available out there in the market? Can you please give us a little bit overview on that?
So far, I have heard of Pfizer,
Yeah.
Moderna, AstraZeneca, and also Novavax.
Yeah.
The difference is Pfizer uses encapsulated mRNA vaccine, so
Yeah.
it is using, like I said, messenger RNA. Its efficacy rate is very high, 95%, I guess. And the Moderna also uses the same approach, like
Yeah.
encapsulated mRNA. Like this is also
Yeah.
an encapsulated mRNA vaccine.
Yeah.
So what they do is like coronavirus has spike proteins on them.
Yeah. Yeah.
So the
Yeah.
crown-like structures coming out. This is
Yeah.
what it uses to attach itself
Yeah. To the
to
body.
the
Yeah.
human cells. So
Yeah.
it it is basically messenger RNA encoding that spike protein of coronavirus has been used. So the body recognizes it, and then the immune
Yeah.
cells recognize it and build a response.
Yeah.
The AstraZeneca one that is developed by Oxford is a
Yeah.
viral vector vaccine. So
Yeah.
it's using double stranded DNA. You
Yeah.
for the spike protein is being used in
Yeah.
it is, I think it has been transferred to a safe virus mode.
Yeah.
So expressing the spike protein.
Yeah.
On the other hand, the Novavax that is also developed in US also
Yeah.
uses virus-like particle vaccines.
Yeah.
Yeah. So there are different approaches.
Yeah.
The one from India
Yeah.
is the SARS-CoV-2 actually itself that is chemically inactivated.
Oh, okay.
Chemical. So
Yeah.
it can replicate, but the proteins or whatever information is required for the immune system to get
Yeah.
a response is
Yeah.
present in there. So they use a chemical to kill that
Yeah.
virus and
Yeah.
that's it. So that's the one from the India is making.
Yeah. So are all these vaccine equally effective, or there is a difference in efficacy of these vaccines?
So looking at the news, what I hear is
Yeah.
Moderna or Pfizer is more effective
Yeah.
compared to AstraZeneca one.
Yeah.
And also I can see that, but the beauty about AstraZeneca one is relatively very cheaper, and it can
Yeah.
also be you can store it even in a regular freeze temperature. However,
Mhm.
the Moderna and Pfizer ones you have to
Yeah.
store it at -70 because
Yeah.
it's a messenger RNA that gets degraded very fast, right? So also the cost of manufacturing is higher.
So these messenger RNA vaccine are fast to develop but difficult to store.
Yeah, I think in the long run, people, scientists or researchers will eventually
Yeah.
come up with solutions for this as well. But I believe that is the reason why
Yeah.
you the storage temperature is also a hazard,
Yeah.
especially for the Pfizer one. But
Yeah.
they are highly effective.
Yeah. So, Kanchan, when you mentioned that AstraZeneca, I remembered the recent news. There have been a lot of news going on about the side effect of this vaccine. So, what do you think about that? Can you elaborate a little bit on that area?
I think the efficacy trials and the data that I have seen from AstraZeneca or even Pfizer and Moderna, they are all good. They are highly, the efficacy is very high, like
Yeah.
90%, 95%. So they are all
Yeah.
good. The risk of blood clots has not been totally, like it has not been totally linked to the use of this one.
Yeah.
Use of AstraZeneca, there are cases reported in some countries. But
Yeah.
if you look at the overall picture,
Yeah.
there is a 0.0004% chance of having a
Yeah.
risk of blood clot when
Yeah.
you get immunized with AstraZeneca vaccine.
Yeah. Yeah.
However, the chance of getting blood clot if you don't get a vaccine and get COVID infection is,
Yeah.
I think, more than 15%.
Yeah.
And at the
Yeah.
same time, like if you take a birth control pill or if you
Yeah.
smoke, the chance is already 0.1%. So that's
Yeah.
1,000 times higher chances
Yeah.
for other things. So, and it's not, the study is not only from 1,000 people or 10,000 people. It's
Yeah.
from millions of people. I mean, UK has already administered like more than, I don't know. I don't know the exact number, but like millions and millions of doses, and so have so many other countries. Like even
Yeah.
in Canada, the health minister of Canada got the AstraZeneca vaccine, and even
Yeah.
the premier of Ontario.
Yeah.
Like that is to boost the morale of people as well. Like there's nothing wrong with it.
Yeah.
However, I would like to add that Ontario Canadian government has just taking this into consideration that there could be a tiny bit of chance that it
Yeah.
could happen. They have not. They have stopped using it for people below 50 years of age. So 50 will get AstraZeneca
Yeah.
vaccine,
Yeah.
but in UK, almost everybody is taking this. So this is a good vaccine, and I would advise all the people to go out there. If there is a chance, please go get vaccinated because that's the way we can defeat this horrible
Yeah.
disease, and we can hopefully lead a normal life again
Yeah.
very soon. Otherwise, the more people get vaccinated, the thing of herd immunity and everything comes into play, right? But
Yeah.
if there are still people who is not vaccinated and it keeps on spreading, so
Yeah.
I think that we all need to do our part and
Yeah.
need to, if possible, we need to get vaccinated to get safe from this.
Yeah. So as of now, vaccine is the only way going forward.
As of now, for coronavirus, yes, of course, vaccine is the only way going forward. However, it's a very interesting question because now I can talk more about what I have been recently involved into as well. There are
Yeah.
different approaches. So, like now, a lot of research labs are also trying to develop antiviral drugs instead of vaccines.
Yeah.
So, to develop these drugs, what will, like, for example, our research lab, like looking for or designing drug-like compounds and testing them to see if they can actually stop the replication of coronavirus. So uh, there are these two different kind of proteins in
Yeah.
coronavirus.
Yeah.
that they use. So NSP14 and NSP16. So
Yeah.
what they do is when the spike protein binds to the host cell to our cell
Yeah.
in our body, what it
Yeah.
does is the messenger, the job of this protein NSP10, NSP14 and 16 is that it mimics our immune system pretty
Yeah.
fast,
Yeah.
and that's how it evades the host defense system.
Yeah.
So what we're trying to do is like we're trying to stop the activity of this, like to inhibit or stop the activity of these two different enzymes in our lab.
Yeah. Yeah.
And similarly, there are other non-structural proteins of this coronavirus, and also people are trying to develop
Yeah.
drugs to stop this binding of this spike
Yeah.
protein as a whole as well. So a lot of research has been going on, and the beauty of antiviral drugs is once you get and this most the sequence of these non-structural proteins, for example,
Yeah.
14 and 16 and other proteins, are so highly conserved between all
Yeah.
the coronaviruses that are possible here. So even between SARS or MERS or even this one and all the others that were there is highly conserved. So once you find a molecule or like drug against this,
Yeah.
it will basically stop not only the ones that were prevalent before, but ones that might come even in the future. So
Yeah.
uh, a lot of effort has been going on with collaboration with university, and different companies have also been heavily involved.
Yeah, so these antiviral drugs and these vaccines, nowadays there is a lot of mutation going on in the coronavirus. So, do you think these antiviral drugs and these vaccines will be able to fight against this mutated strain as well?
So I mean, we do because there is no antiviral drug for as of now, so we do not know,
Yeah,
right? But
yeah,
so that's the problem for vaccine is these virus are they get mutated very fast. That's one of their
yeah,
characteristics that they mutate. They keep on mutating. So that's why we do not have any vaccines or any treatment for
yeah,
HIV, right? Because
yeah,
it keep mutates so fast. And similar is the case now that with different variants coming, it might the efficacy or efficiency of the vaccines might go down. So the approach that researchers are using these days, like we are using, for example, for developing antiviral drugs, is that we targeting the portion of the virus that
yeah,
is conserved like throughout the species.
yeah,
So no matter what mutation it has, the sequences of these non-structural proteins are until now are like almost 99% or
yeah,
90% similar. So
yeah,
once you target that and you make it inactive itself,
yeah,
if there is a compound or drug or medicine that can actually target those portions,
yeah,
then what I believe is like even though there are mutations, it
yeah,
might not affect so much.
yeah.
So that's why, as I mentioned before, it could even work for many different coronaviruses that could emerge in the future as well.
Yeah, that's great concern. So there are still many people out there who are against the vaccine or who don't like to get the vaccine. What do you suggest to those people?
It is personal choices. I respect everybody's choice, but if it was, I would request if it's possible to please get vaccinated. Like I said before,
Yeah.
we need also herd immunity, right? So
Yeah. So Kanchan, thanks a lot. Slowly we are coming towards the end of today's video. Thanks a lot for sharing your expertise. So before closing out today's session, any final thoughts you would like to share to our viewers?
thank you, Biso and Winspire, for the time. I'm happy that I get a chance to express my few words. Although I'm not that much of an expert on vaccine or something, but I have been closely following this because I've been working with drug design for this COVID now.
Yeah,
So thank you very much. And yeah, my advice is please stay safe, and together we can defeat this disease.
yeah,
And if possible, please get vaccinated. The benefits of vaccination like outweighs or like is way greater than the
yeah,
harm it might cause. So
yeah.
yeah, please get vaccinated. Stay
Yeah.
safe.
Yeah, that's great. Thanks a lot, Kanchan. So guys, I hope you liked the content of today's video, and I hope you like the content of my channel. If you like the content of my channel, please like my videos, comment down below in the comment section, and also please do not forget to subscribe to my channel. I'll see you again in next video. Until then, bye. Thank you.